Good morning, afternoon, evening, chameleon wranglers. We have people from all over the world. Jenny, you made it. Hey, we got Jenny from Germany. So we do have
Yeah,
Europeans here.
exactly. I thought everyone would be watching the football.
And for those who don't know, this is Dr. Mark Scherz. Mark, thank you for coming on.
Hello, it's my pleasure to be here.
Okay, so what's going on with all of our European friends?
Well, there are two things going on. On the one hand, a lot of them were at Hum today, the world's largest reptile expo or exotics expo, and on the other hand, a lot of them are going home as quickly as they can so that they can watch the Champions League final, which fair enough. So.
All right, Jenny, you're gonna have to put in the chat. Give us a little summary as to what happened and what you saw in Hum, especially chameleons. Mikey, hello. James is here. Hello, James and Two Turtle Tom. Oh man, we've got a great crew here today. So anyway, I'll while Jenny is giving us the show summary, go ahead and give you a background as to what we're doing here. Um, Jenny says, "Cool, could not care less about soccer." Oh, she's using the American word for it. Thank you. Hello, Stephen, Natalie, and so we're Kaluma Brevicorn is in the news today, the chameleon news, because we have some people in the community working with Kaluma Brevicorn, and they needed a care guide, and there's no care guide. There's little bits and pieces of Kaluma Brevicorn, and it's very difficult putting together a care guide on something that it's not a whole lot of people have experienced throughout the entire life cycle. And so to put together a care guide, I have to go and I have to get pieces from all these different places to put together enough of a life cycle so we can put together a care guide. And so the community can grow. And as we learn about this species, we update the care guide. It becomes a living document, blah, blah, blah. Community grows, Brevicorn becomes famous. So now there's one thing about Brevicorn that is a real pain in the butt is when you get Brevicorn imported in, you can never be sure that you've got Brevicorn. And
Yeah.
so this is a common issue with a number of chameleons. And so for today's discussion, I want to bring on Dr. Mark Scherz, who is a real life chameleon field scientist. He's been in Madagascar. Madagascar's tried to kill him a number of times, and he prevailed and is here to talk to us today.
Yeah, that's true.
Oh, let's see. Hey, we got someone from Isabel from Montreal. Hey, welcome, welcome, Joanne. Oh, Joanne's from Canada too. Our Canadian people are coming in. So, all right. So, Mark, what's up with this species? Why is it that I guess maybe give an overview of the complexity of Brevicorn? Why is this giving us so much trouble?
Yeah, well, so first let me give a little bit of context for why I know anything about this group, I guess.
Yeah, that's a good idea.
So obviously, like you just said, Bill, I've been working in Madagascar for a really long time. Depends when you kind of start, but since like about 2009, I guess you could say I've been working more or less with or in Madagascar. And over that time, I've done on uh, various different expeditions across different parts of the country, and obviously I've worked quite intensely together with David Prutze from also from Germany, and Frank Glav and Miguel Vences. So all of these brilliant people working on the chameleons, and we have paid most attention to the Kalama Nasutum species complex, because that was the one that needed the work the most. But throughout my expeditions, I've gotten to know various different members of the Kalama Brevicorn complex at the same time, and many other aspects. And the Brevicorn complex is, I think it's a lot bigger than most people realize. And it is challenging, let's say. Hi, Patrick. Yeah.
Oh, I say, sorry, we got our friend Patrick from Madagascar.
Yeah, super. So the Columba Brevicorn complex is really challenging because it's a very large group of species, and I think in a little bit we can go through all of the different species. We can talk through each of them, and because I think most people will have heard of Columba crypticum, Columba Brevicorn. If we're lucky, they might have heard of Columba Maita, or maybe even Columba amber. But there are like 7 more species that you might not be familiar with. And they are added to that mix. It's a very interesting and complex mix of things that's going on in this group. And from a taxonomic standpoint, from the standpoint of the people who try to understand these things and like figure out how many actual evolutionary units, how many species do we have, and where are the borders between them, and how do you distinguish them, and those things. Things are even more complex than people who are just trying to keep them are aware of too, and we'll get into that, especially as it pertains to Columba crypticum, which is yeah, we'll talk about it. So
Okay. And
whenever
by the
you're ready,
way,
we can.
anytime you want me to bring up
Yeah,
the slides that you know I have, you just let me know.
yeah. Well, I mean, perhaps we can do that in a second. I think you might want to catch up on the great stories here we're
Yeah.
hearing
Okay. Yes.
from the Hum show.
All right. We do have our summary. It was hot. Tons of people. Smaller than usual. Let's see. She saw lots of panthers. Didn't see many veiled. Oh, come on. Veiled are so nice. But I met a nice breeder of Hunelli.
Yeah.
Mark, how would you pronounce that?
Hühneli. So I would pronounce it the German way because I learned most of these names in German. So
Hunelli.
when basically you should look for the name. If it ends with an I or it ends with an AE, you should look for the name. So if it's Hühnel, after whom it was named, then you would say Hühnel, the way Hühnel would be pronounced, and then you just add the E sound. So it would be Hühnel E.
I like it. Okay. I'm going to practice that. That's how I'm going to say it from now on.
Yeah. So if you're talking
Hunelli.
about Glavi, it's Glav E because it's Frank Glav
Okay.
E, or Vences
Hunelli.
E. So that's the easiest way to go around with those names.
All right, everybody. You heard it here.
Crested gecko for 7,000 euros. Excuse me.
Wait. What? Crested gecko for 7,000. Oh, okay. Wow. I'm
Holy
glad to see
smokes.
that there's somebody's making some money out there.
That is a crazy amount of money. I've only ever seen that on a ball python. Wow. Well, and of course, lychees and whatever.
Halli hallo. That sounds German.
It does sound German.
All right, Sarah, welcome.
Cool.
And okay, so we have an explanation. It was a black body with a white tail. All right.
Okay.
All right. That
Yeah.
I guess that's what they love over there.
Sounds like a fluke that's not breedable, but you can make a lot of money off of it.
Sean's going to take 2 of those. So,
Yes. Yes.
oh boy. Well, oh yeah, and everybody, we got to say, this is Patrick from Madagascar. There was Patrick and I had many conversations with Mark while we were over there about these species, and so this is just a continuation of that discussion.
Yeah.
And there we go. We got someone from Holland. Yes, the Europeans are making a show.
Lovely.
All
So,
right.
Bill, whenever you're ready, we can start to dive
All right.
into
Let's
the
do
full
this. See,
group. Shall we do it?
let's do it. See, everybody, this is what goes on whenever, like, so people want to start a Brevicorn breeding group. The problem is these things look so alike that it's a challenge to verify that you have everything is of the same species. And so this is a slide. This is an example of this was all taken in Madagascar when I was in Madagascar. and these are different species. And so, Mark, when you see all this, what do you see?
So, I mean, there's a couple things you need to be looking at when you look at any Columba species, and the first thing you need to try and do is figure out the sex that you're looking at, because that's going to make a really big difference. And for various historical reasons, um, females have been given far less attention than males, both because they lack those what we call secondary sexual characteristics, those strong. There's always there can often be strong um, dimorphism in the ornamentation, the coloration of chameleons. The males and the females have different appearances. So dimorphism is shape, and dichromatism is the word we use for two different colors. And in the case of various different Columba species, it's quite pronounced. And as for historical reasons, the people working on the taxonomy of things have paid most attention to the males. And so we do not have particularly good information about the females. I just want to have a shout out quickly to say hello to Desiree Schumann, who's one of my students, actually. So
Oh,
Desiree
really?
is joining us as well. That's lovely.
Hey,
Um,
hello, welcome, Desiree.
so Desiree is working on some frog skull segmentation things. Is just trying to work with the very large skeletal anatomy data sets that we have. Lovely to have you here. Yeah. So as I was saying, we have pretty good tools for figuring out the identity, the taxonomy of these species if they are male. If they are female, things are right troublesome. So, looking at this picture, the first problem that we are faced with is that we're trying to identify four female chameleons. And within the Columba brevicorn group, the females are extremely similar to one another. So similar that in some cases, the field guides, etc., essentially say, don't bother, you can't do it. So, now we have those animals coming into the pet trade. We don't know where they're from. We don't know how they got here. We don't know anything about, and even if they came from one place, you might have four different members of the Columba brevicorn species group in a single location. And now you have this problem of how do I breed these? How do I figure out, am I creating hybrids in the process? Because a lot of these things will hybridize and or they may hybridize, and they may even produce fertile offspring. That doesn't mean that they're not species, because we've moved beyond that as a concept for our species. But this is a serious problem. So talking about these pictures that we're looking at here, first of all, yeah, these are four females. You can see that there's variation in how strong that lateral white stripe is. You can see that on the top left picture, it goes through those paired white markings with the black outline. Those are typical of the entire. In fact, most Columba species, not just the Columba brevicorn species group, but most Columba species, if you include the Nasutum group as well, have markings similar to that, right? So those paired white dots and the black mark or the dark markings around them are very typical of Columba, and then you have that white stripe that is sometimes there. And of course, like chameleons do, the color comes and goes. What we have to say about the top right picture is this is a sneaky one, because this is actually Columba Maita. Now, how do I identify it as a member of the Columba
Now, when you call it might.
Malte,
Oh,
Malthe,
okay. So we in America call it Malthe. So if everybody,
Malthe. Yeah,
but you go ahead
sorry.
and say it the way you say it. I just
The
want to let everybody know. I'm going to
yeah,
do the translation.
Malthe or Malthe or Malte. I mean, if you're being German, you'd call it Maite.
Okay,
So
you do it that way.
so my Kalama Maita is an interesting case. So we have this group of chameleons that are fairly large bodied. They get pretty big, not including the tail. We're talking about animals that are maybe 20 centimeters. Bill, if you want to do the inch conversion, you're very welcome to. But let's talk in centimeters for now. So 20 centimeters, maybe even a little bit bigger than that. And some of the species are pretty small. So Columba taratananense, for instance, is a pretty small species. So there's a lot of size variation. But these ones are not the very thin species, we could say dorsoventrally narrow, thin species of the Columba Nasutum species group, where we have very different morphology there. These are larger, higher bodied, deeper bodied species, and they all have flaps on the head. But flaps are not diagnostic of this group because there are flaps in the Nasutum group, and there are flaps in the Parsoni species group as well, because Caperoni has flaps, and various other members have a little bit of occipital lobes, what I'm calling flaps now. Right. So Maita has an elongated head, and as a consequence, longer occipital lobes. It has various other features. It also has a pretty strong, in males, very strong rostral appendage, that nose piece at the front. So Maita is an interesting member of this group, but we'll get back to that problem because Maita is actually not a member of the group. We'll get to it. So the top right picture, that's Maita. You can actually tell because of the angle of the occipital lobes as well as other features of the head. The rest of these, they are from Fianarantsoa. So Fianarantsoa, or Fianar as we say, is just west of Ranamafana. Ranamafana is in part of Madagascar that we call the south central east of Madagascar. And when we talk about Columba crypticum, I would say 90% of observations of Columba crypticum come from Fianar and Ranamafana, coming from that area, which is important because of what happens next. Taxonomically, Columba crypticum was described not from that area, but from a little tiny fragment of forest northwest of Antananarivo called Ambuitantili. So Columba crypticum is a species that is described from the central highlands of Madagascar, and it has an extremely short snout in the description. It's actually characterized in the description by having a snout that is less than 4 millimeters, exceeding the front of the lip.
Right? Now, is that one up in the northwest, was that actually the Crypticum that we recognize today that's from down
we don't
south?
know.
Oh, okay.
So this is where it gets fun. We know if you go on iNaturalist, you can find observations of this Columba crypticum, the real Columba crypticum, as it was originally described by Chris Raxworthy and Ronald Nussbaum in 2006. Okay. And it is characterized by a very, very short rostral appendage. Now, Bill, do you have a slide that shows some male Columba crypticum? So,
yeah,
Columba crypticum, that left picture there, you can see it has a massive rostral appendage, really long.
Yeah, it was impressive.
Yeah. So that 4 millimeter line, let me just make sure that it was 4 millimeters, but I'm pretty sure that it was. Anyway, they're characterized as having a very, very short snout. They do not show that outside of Ambuitantili. And now is the question. Do we have Columba crypticum being observed at all? Or is Columba crypticum only in Ambuitantili and everything else is an undescribed or possibly described, that's another problem, species? So This throws even more problems in the mix, but only from a taxonomic perspective. It doesn't matter for the people who are breeding them because it's very, very unlikely that animals that are coming into the trade are coming from Ambuitanteli or other forests in the Central Highlands for one very good reason, which is that there is no more forest in the Central Highlands other than this very strictly protected tiny, tiny fragment and a couple other ones.
Okay, well, I guess that's convenient.
Yeah, exactly. So we will, for the rest of our conversation, for the most part, pretend that we are applying the name Crypticum correctly. And we can ignore all of this kerfuffle for the time being.
Okay.
It will eventually be clarified, but at the moment, all it really does is say to us, there is a bigger mess here than meets the eye, in the same way that the Nasutum species complex was a much bigger mess than meets the eye. And actually, recent results that are not published make it even worse. So, there, like the Nasutum complex is one where you can lose a lot of hair over. The Brevicorn complex actually, or the species group, actually by comparison, is manageable, let's say. Okay.
Okay. Now,
So, yes, Bill.
well, okay. Should you finish your thought before I bring in another, or is this a good breaking point?
This is a perfect breaking point because
Okay.
my next thing was going to be to embark on a brief explanation or background on the rest of the members of the group, just so that people get familiar
Okay.
with
All right.
the rest of
So
the group.
I'll throw in. You mentioned, and this is the big problem that we have, is that when we get specimens in, it's hard to identify them. And then this isn't because somebody is trying to smuggle or do any, I mean, this is an honest mistake of people saying, okay, this looks like Brevicorn. I have quota for Brevicorn. I don't have quota for Crypticum. So I only want Brevicorn. How much overlap is there with all of these species that look alike? And what species are involved in that overlap?
Yeah, so that points toward part of this problem. I'm going to assume that the vast majority of collecting for the pet trade is happening in the rainforests that run the eastern belt of Madagascar.
Okay.
And I think I have pretty good grounds for assuming that because the pits in the north are challenging to access at the best of times, and there are all kinds of problems up there. Very few species come into the pet trade except, well, from that area, except for things that come from the far north in Montagne d'Ombre, which is another problem, actually. It could even be that we are having animals coming into the pet trade. This one behind me, this is Kalama Amber from
Mhm.
Montagne d'Ombre, and it looks damn similar to other members of the Kalama um, Brevicorn species group, right? So they might be coming in as well as Brevicorn and being mixed in here as well. So in general, there are often a couple of species in this complex found together. Um, it's not always super bad because Maita uh, is the most frequent second species. So Kalama Maita has an enormous distribution in eastern Madagascar and northern Madagascar. It basically spans, I think it even goes as far south as, one second, I can confirm this without saying it wrong.
Okay.
So Maita goes, it's as far south as the far southeast tip of Madagascar, down in an area called um, uh, well, it occurs close to Tuamasina, so down in the area around Andoahela, basically. So very far southern tip of Madagascar, and then it goes all the way up the east coast, and then up into places like Marajeji, and goes all the way across northwest to Tsaratanana. and also into Serrata. So it doesn't seem to occur on Montagne d'Ombre, but that's basically the only place that you don't
Okay.
find Kalama Maita in the rainforest. All right. So Kalama Maita is everywhere at the same time as basically everything else. There's another really, really widespread member of this group, we'll get back to it, which is Kalama cuculatum.
Oh, I love cuculatum.
But cuculatum, one of the most beautiful species of Kalama, in my opinion, Kalama cuculatum is super widespread, but everywhere it occurs seems to occur at densities of practically zero. So finding them is a huge fluke. And I don't think I know anyone who has encountered, or I haven't heard stories of anyone encountering more than one on a single trip, which I think is
Okay.
remarkable. So Kalama cuculatum is a curious beast, and keeps popping up in places where we wouldn't expect it. So it's also often in the same place, right? Um, so that would be three species throughout, where Brevicorn is, there would be these other two, and then where Crypticum is, as we call it, Crypticum, in further south, probably there is also Cuculatum and Maita, right? And then we have other species being added to the mix, which we can get to after this brief update of things from the chat.
Oh, okay. Yeah. Well, let's go ahead. Thank you very much for. We have one question about. Um,
Yes.
I'm going to have to let you.
So this question says, are there animals to be found in the Brevicorn group in the Anzanaharibe Sud? So in Malagasy, the J is pronounced as we pronounce a Z, so it's Anzanaharibe. And the answer is yes, absolutely. There are multiple members of this species group in Anzanaharibe Sud. The most frequent that you're likely to encounter would be Kalama Maite. I'm just trying to think if there are other obvious ones that could show up there. There's a decent chance that Kalamajeji could be there, which would be interesting. It's a high peak endemic, so it would only be at the tippy top. The same is true of Kalama Peltierorum, which is a bit of a weirdo. And I think that would be it. So it would be basically those ones. There's a decent chance that you would also have Crypticum or Brevicorn in that area too. So
Okay.
not Crypticum, Brevicorn in that area too.
So this is a observation. We saw them near the road. Are these?
Cuculatum?
Well,
Could be.
I'm not sure the context of when this comment was written, so it could have been at any time we talked about these species. But being there, I know it was very easy to see these Brevicorn on this as we were driving by. Stop the car. Okay, there's
Right.
there's a Crypticum. Are they deep forest or are they forest edge? What's up with this?
They are yes.
Yes,
Is the
that's right.
answer. They are both things. So all members of the Brevicorn group are true deep forest animals. But there is this thing called the edge effect, where many species occur at very high densities, like weirdly, anomalously high densities, close to the edge of the forest. I can't explain it in any detail, but in essence, the edge creates this area of high diversity, density, possibly because you have a combination of things that are going through that gradual succession that happens at the edge of a forest as it kind of changes. Um, and we very, very frequently you find chameleons along paths in forest, and that's not probably because you just happen to be encountering them, but there is, I think, an elevated chance of encountering them along those paths. Yeah.
I
So
was assuming because the edge of the forest that gives you access to sunlight, it gives you access to shade. With the
right.
sunlight comes flowers and pollinators and more
Right.
food.
And you have to bear in mind that when we're walking through the deep forest, we are not walking where chameleons want to be, because in, logically, if the chameleon wants a lot of shade and especially a lot of sun, then you're walking through the understory, but they're walking in the canopy and they don't care.
Yes.
So we don't know because we can't observe.
Mhm.
But yes, and there have been studies on I think Trioceras species in mainland Africa, where they have looked at this. What is it about the edge that they're using? And it's a combination of all these things. You have the light changes, you have more insects doing things because insects also like to sun themselves and then get zapped by a chameleon. They don't like that part, but they like the first part. And uh, so it makes for a whole series of different things that's kind of driving things toward the edge of the forest.
Okay. And then we have,
Yeah.
all right,
So,
nice meaty question.
right. Okay, so basically, Columba varicosus. This takes us completely away out of not
Wait,
Columba
wait.
varicosus, of
Yeah.
course. Furcifer
Oh, don't
varicosus.
be doing
I,
this.
I have
That
to
that would
change.
be
I
more
have
of a
to
change
change
than we could
gears.
handle.
Yeah. No. It also gets confusing because of Columba gularis, which is a name you probably haven't heard of. So, Furcifer varicosus is a beautiful species found in dry forests of western and southern Madagascar. And there were results that came out in a paper in 2016, which I have sitting in front of me. That paper is titled A Phylogeographic Assessment of the Malagasy Giant Chameleons, Furcifer varicosus and Furcifer ustuleti. This was written by Antonia Florio and Chris Raxworthy. And in this paper, they looked at a lot of genetic data from Furcifer ustuleti, which is the giant chameleon, which Bill, I believe you have a slide of actually.
Oh, yes, I do. We can.
That brings us up nicely. So Furcifer ustuleti is the world's longest chameleon. And so when it comes to our battle of what's the largest chameleon in the world, Madagascar has both the longest and the heaviest, but it's not the same species. So Furcifer ustuleti is the longest chameleon in the world. Large, heavy animals. They have that massive casque. And note, Bill, your picture on the left there is absolutely perfect for this. Look at the shape of the casque, that domed shape to the casque. That's quite typical of Furcifer ustuleti. In Furcifer varicosus, that curve is straighter. It's not as pronounced, and the dorsal spines are also much larger, much more pronounced. Varicosus is gets that name. It's called the spiny or warty chameleon. It has more spines and larger tubercles over the flanks, but then basically what distinguishes these two species, the easiest way to distinguish them if you want to do it in a museum, is to count the number of spines on the dorsum, and the numbers are what differentiates them easiest. And so that's the difference between ustuleti and varicosus. Then comes the question of what about these two different clades? So in this paper by Antonia and Chris, they found two major genetic groups of Furcifer varicosus and two major genetic groups of Furcifer ustuleti. Which, of course, is not that surprising. These are animals that are very, very widespread. Ustuleti occurs across basically all of central, western, and much of also northwestern Madagascar. Even northern, the far northern tip of Madagascar. And then varicosus, basically, if you cut Madagascar in half and then cut kind of diagonally through it, varicosus is found south of that diagonal line across all of that kind of heathland, shrubland, etc. Grutemark, we will get back to the Columba Peltierorum thing in just a minute because that's a big part of this the Columba Brevicorn group. So varicosus is found south of that line, but in the results from this paper, this 2016 paper that was published in PLOS One, which is an open access journal. Anybody can go and read this paper, although you might not enjoy it if you're not used to reading these kinds of papers. Varicosis is found to be divided into two groups, and those two groups are divided essentially into a southern group and a northern group. Now, to my knowledge, so they did a little bit of niche modeling here to find out what the niches were like for these two things. But there are some issues. Firstly, there are locations where if you draw a kind of a line around all the points, there are locations where both would be found. So the niches definitely would overlap because the space definitely overlaps. But in general, they found suggestions that the ecological niche that these things occupy, so basically the kind of environments they were living in, is, different to some extent. But in this paper, they basically didn't find any substantial differences in appearance of these animals or morphometrics between these animals. So to quote the paper on page 12 out of 23 of this paper, based on the characters measured and scored, there are no clear morphological distinctions between the genetic clades, so potential cryptic species, found within each species. So that is both for Varicosis and for Usuleti. In both cases, there were these deep clades, right? So all that to say, what distinguishes these clades from one another is genetics, and that's it. What distinguishes the northern Varicosis from Usuleti is that the northern Varicosis still has the traits that make it Varicosis. It has fewer, larger dorsal spines, and it has a slightly differently shaped casque, especially in males. So that's the answer to the question. That was a really, really long answer, but that's the answer to the question.
All right. So let me summarize. Are you saying there isn't a difference?
There is no difference morphologically between, basically, within Varicosis and within Ustelleti, there's no grounds for treating the populations that are genetically different as different species.
Okay, now how does it affect those of us like Gabriel who's trying to establish them in captivity? How much do they have to worry about animals being brought in from different areas?
I honestly wouldn't worry about it. You might find that there are things like behavioral isolation, so these things simply won't mate with each other. They might have Antonia and Chris in their study, they didn't look at the genitals of these things, but chameleon hemipenes, so the reproductive organs, are covered in spines, right? And often, very early in evolution, those spines will differ and they might not work or fit anymore with females of the other species. So it could be that there is something already arising that we wouldn't be able to tell. Or they can maybe mate just fine, but the eggs die in situ or whatever. This hasn't been tested. So it's basically a risk that you have to undergo in the same way that you essentially have to undergo risks when you're bringing in females from genetically separate populations of Pardalis, and males, and you don't know if they're actually from the same location, even if they come in with being from the same location. Those tests are making themselves in a way because you have no other way of gauging it.
All right,
you could sequence it, but that's a lot of money and faff.
Okay, is there a possibility that there is can I say a cultural aspect to it? If, say, genetically they're similar, but they're isolated, could chameleons get to the point where they are the way they communicate their mating?
So we wouldn't call that culture because culture in biological terms is very strictly defined. Culture is basically non-genetic effects that are passed on through learning, and these behaviors are unlikely to be learned because of chameleon behavior in general. So rather we would just say that they become behaviorally isolated. So there are these changes to behavior. It might be that the males, if you think about birds, the male's song and dance doesn't fit what the females are looking for. And so you could have things where all the posing and posturing he likes is never going to seduce the female. Of course, chameleons are coercive maters, which is the gentle way of saying that the female doesn't have a lot of choice. But she does have some choice, and females are aggressive toward males that they are rejecting, and they can successfully prevent unwanted copulations. So I would say that in general, yeah, it's not unlikely even that there would be those kinds of things that develop, but it would be something that people might only observe by chance, or they might simply, often when you put two animals together, they'll reject each other whether they're from the same genetic population or not. It doesn't matter. They just don't. They're not compatible somehow, right? So it's very, very hard to infer unless we have genetically identified populations. That's basically what I would say.
Okay. We'll do this question while I try to recover from breathing in my drink.
Yeah. So, sorry, Bill. The genetic variance between the species is substantial. So we don't really talk about genetic variance between them. We talk about genetic distance between them. Now, the genetic distance between members of the Brevicorn species group is substantial in the same way that genetic distance among members of the Nasutum species group is substantial. So these are differences that are enough to make a difference, or are enough that we might start to suspect that these are separate species. That's also true of these deep lineages within Furcifer vericosus and Furcifer ucelleti. I'm not sure which of these groups you were referring to, but in each case, these are cases where people are raising the issue that they might be separate species because the genetic distance is substantial. Bottlenecking is kind of a different phenomenon where through population collapse or maybe colonization of a new area, you basically have less genetic diversity, and then there's fewer genes to go around. And in essence, we don't have a lot of evidence that that's happening in anything. That's mostly because we haven't looked for it, because we don't have the money to look for those kinds of things.
Okay, it's now a good time to start talking about
Yeah.
the species.
So let's talk about the different members of this species group. Okay.
All right, this is something that many of us, including me, I have not heard of this species before. Can you give us a background as to what is this?
So we'll get to Peltierorum as we go through. I think
Okay.
I'd like to address them in the order that they're given by the Glavenvense's Field Guide to the Reptiles and Amphibians of Madagascar, the 2007 edition. So that's the latest edition, which of course is now many years out of date. But fortunately, no species from this group have been described since then. And I'll just do it in that order because it's the one that I have in front of me. So we have this guy, Columba amber. Columba amber is a very similar species to Columba brevicorn. It is only found on Montagne d'Ombre and possibly in pockets of forest nearby, but I'm not entirely sure, and I don't have my annotated copy of the field guide with me tonight, so unfortunately I can't say for certain what the situation is with Columba amber in other locations. But anyway, Columba amber. This species, the males are this kind of orangey, yellowy thing. They can also be pure brown, boring things. They can, when they get fired up, one second. Where's my settings thing? I can show you what they look like when they get fired up. Here we go. Virtual background. This is the one. Can you see that? I don't know if you can see that. This is a
Yes,
fired
we can
up.
see it. Let's see. I'm going to see if I can bring up.
Oh, now your face is right in the middle. There we go. Okay, so so this is a fired up Columba amber. right? So you can see those orange colors on the head. You can see the kind of green on the snout there. This is a male. You can see he has a relatively short rostral appendage. Okay, so these are kind of general features that typify Columba amber, but in general, they're not super easy to tell apart from Columba brevicorn. Females, I don't have a picture that I can easily show you right now, but females are essentially pure green to brown. Okay, so that's amber. It's located only in the north. Now, the reason we have to worry about Columba amber coming into the trade is that it's from the same place that Columba linotum is found. And Columba
Okay.
linotum is definitely coming into the pet trade. So if Columba linotum is being collected, there's no reason to suspect that Columba amber is not also being collected, right?
Okay.
Okay. So so that's the situation with Columba amber. Then we have Columba brevicorn. Now, Columba brevicorn is a species that we're all kind of a little bit more familiar with. It is originally described. So things get real complicated real quick. Columba brevicorn was originally described from I think from Madagascar. So this species was described in 1879 by this guy called, I think, Alfred Günther. And Günther would have received the specimens from a trader or a dealer or a colonial person living in Madagascar or traveling through Madagascar. Okay, so he got this species. It was from, I believe that it came actually from near Antananarivo, and possibly close to this forest called Sarafidi. Oh, I can figure this out in 1 second. Actually, give me just a moment. Yes. So I have all the things in front of me, so I can look them all up. So Brevicorn was originally described, near Antananarivo in Madagascar, right? Near Antananarivo, extremely helpful, useful information, right? But there were other species there are other specimens in that shipment. One of which is a species that Günther also named, and he called it Chameleon gularis. Now, the trouble is, gularis was a juvenile individual, and not just a juvenile or subadult, but also a female. So, Brevicorn he was describing was an adult male, and then he had this smaller other thing that was a female. Now, if it's from near Antananarivo, where is near Antananarivo?
Okay.
Ambuitantili, where Columba crypticum is from, is also near Antananarivo. And at that time, there would have been forest that was much more widespread across that area. So, is Gularis really different? Is Gularis the same thing as Brevicorn in the first place? Is Brevicorn the same thing as Crypticum? All of these questions are very, very, very hard to answer. But for the average person, don't really matter because it's only going to change names. It's not going to change the species.
Okay.
So let's talk a little bit more about Brevicorn itself. So Brevicorn is this species we frequently see pictures of, and there are a couple diagnostic things. It usually has a relatively long rostral appendage, right? So the males have this substantial rostral appendage that is in several cases like red, dark red color. And these are also relatively sizable chameleons. Yeah. So in this case, that Tuamasina animal, that's a Maita, right?
Yes.
And the one in the middle here, I think, is actually Crypticum. And
the one in the middle
animal,
the one in the middle is supposed to be Brevicorn.
It's supposed to be Brevicorn. Okay, that it could be Brevicorn. Wait a second. Let me get closer.
Okay.
Yes. Okay. Well, I can accept that it's supposed to be Brevicorn.
Yeah, they can't.
so so the distinction between Brevicorn and Crypticum is problematic, right? I mean, that's what this whole conversation is
Yes, yes.
based on. There are these issues about how to identify them. And when you go to the original literature, the basic thing, oh, I said 4 millimeters before. I kept saying 4 millimeters, this rostral
Yeah.
appendage. It's 2.5 millimeters.
Oh,
So 2.5 millimeters is nothing, right? So
yeah.
Crypticum is allegedly characterized by a rostral appendage that is only 2.5 millimeters long or less.
Oh,
So, and the other thing is that the shape of the appendage is supposed to be kind of dorsoventrally flattened, and that the female crest has periodicity, but it actually has periodicity in both species, right? So if you go to the Glavenvensis field guide, identifying or distinguishing these two species is basically impossible, especially when you are finding animals in Fianar and Ranomafauna that we in the community have been, for reasons unbeknownst to me, calling Crypticum, right? So, Bill, maybe it's best if you actually fill in the general community about the characters that are typically used for distinguishing Crypticum from Brevicorn.
well, in the males, it would be because Crypticum, when it fires up, will have blue
It has
legs.
blue legs, right? That's exactly it, right? We call it the blue leg
Yeah.
chameleon for precisely that reason. Yeah.
But when we get into females, so basically, when we don't have the benefit of having a taxonomical scientist to send pictures to, we would say number one, okay, everything that comes in is supposed to be Brevicorn because there's no quota for Crypticum.
Uh-huh.
But then we realize there's so much, they look so similar that the people collecting, unbeknownst, they're not trying to do anything outside of what they're supposed to be doing, end up sending, you look at the one on the left there, that is Crypticum. You
Right.
don't see any blue legs there. Well,
Right.
okay, they don't, the blue legs will come out when they're sleeping or when they're fired up, but a human being comes around and they don't feel like firing up and impressing any female. So this guy gets picked up, and these all get the Brevicorn and the Crypticum get mixed up. And the thing is, yeah, that's the problem. How do we tell them apart, especially when they're juvenile, especially
Yeah.
when they're female?
Yeah.
And the question comes up. Well, they're just a great, from a keeper perspective, Brevicorn is just an incredible species. I mean, they're gregarious, they're hardy, they can take a wide temperature range. It should be a wonderful species to establish. Why are we having so many problems? And it could be, well, we're not pairing up the right animals.
Right. It's definitely possible. So there's some additional information that I can give you from another study. This is a study that was carried out by paper by Boomans et al. In 2007, where they did some genetic stuff. And in this paper, where they did a genetic study on the members of the Columba Columba brevicorn species complex, and in it they identify some major genetic lineages of Columba crypticum. And in 1 second, I will upload a picture as my background because that's actually the easiest way for me to share pictures here. Okay, so yeah, I think you can see that, although it might be mirrored. Yeah, it's going to be mirrored. That's fine. Okay, so prepare yourself for some mirror logic. All right. Um, so what we're looking at here, this is a big phylogeny. This is from that Boomans et al. Paper. And this here says Columba brevicorn. And that word that's backwards there is Andasibe. Okay, so Bill, you've been to Andasi Bay. Andasi Bay is that yellow point there on the
Yes,
map.
I enjoyed Andasi Bay.
A lovely, lovely place. Highly recommended for any tourist. There are a lot of tourists there, but it's a great place to be. This population assigned to Columba crypticum is from Mandraka. Mandraka is that little green point on the map. You actually basically have to drive through Mandraka to get to Andasi Bay. Okay, so these populations, allegedly having different species, or genetically different species, right? They are very, very close to each other. So, and in this study, they didn't have any genetic lineages of Brevicorn from outside of Andasi Bay. So, what we have this problem. All of these green dots here, those are all Crypticum, right? Crypticum is found widespread in eastern, southeastern Madagascar. This is another species we'll get to in a second from Itrimo. Actually, we won't get to that species because it's not described. But the orange one is from Ankaratra. And this is Columba Heleniocy, which is what the question was about. And then, of course, we have Columba amber all the way at the tippy top, right? So lots of genetic variants within and across the eastern mountain chain down to Andawahila in the far south here. And Ambustra, which is also actually Ambustra is up here, so that genetic lineage is quite widespread. So the point being that we have a lot of variants, a lot of widespread Columba crypticum, and then Brevicorn very, very, very close by. Okay, so what does that mean? Well, it means that animals that are coming into the pet trade that are identified as Brevicorn may very well not be Brevicorn, especially females, but males as well. This trait of having the blue legs, I don't know if that's actually confirmed in the populations down in the very far south of Madagascar. These things in Anduahila. I would have to go and try and find pictures from the far south of Madagascar. But to my knowledge, that has not been. Well, I would say I don't know is a simple answer.
Okay.
But at least the animals from Mandraka, of which there are photos in the field guide from of Frank Glav and Miguel Vences. So the Crypticum that are found here have blue legs. So at the very least, it does seem like it is a truly diagnostic feature where it matters, which is where these two things overlap, right? So the males, we can be kind of comfortable in our idea that the males would be distinct or distinguishable. Females are always going to be, or are likely to be, a matter of. I'm just bringing back my Columba Amber background. The females are likely to be a bit of a challenge all the time. And you may simply have to do trial and error in order to find out what they are. In principle, if you know of someone who can do it, and please don't contact me about this, but in principle, you could have your individuals sequenced. And you wouldn't have to sequence for very much. It would not probably, if you did it for a lot of animals, if you were an importer, for instance, or someone who was buying wholesale from an importer, you could have like 50 animals sequenced for $200 probably. It's not that hard, and you could do it from swabs or from tissue, tiny tissue samples, or even cloacal swabs. You could probably use. So it would be possible, in principle, to figure this stuff out directly if you have a little bit of money that you're willing to put aside for it, and can find someone who's willing to do it. People who are willing to do it they might charge more than those $200, but in principle, it wouldn't cost them much more than $200.
Okay. All right.
So we could go on. So shall we go on to some of the other members, the weirdos from the rest, or
Sure.
the diversity of the rest of the Brevicorn group? Okay, so the next species, the reason I say weirdo, the next species in the book is Kalama hafa hafa. So hafa hafa is the Malagasy word for weird. And the reason Kalamahafahafa is called weird is because the rostral appendage, instead of going forward, goes straight up, and it's like it hit the wall. So you will not find good pictures of Kalamahafahafa because basically none exist. But there are, let's say, there is reason to be, how do I put it? Kalamahafahafa is a weird species. It is considered one of the most threatened species of chameleons in Madagascar. It was not, basically hadn't really been seen since the original description until moderately recently. Very, very few specimens exist. The trouble is, the anatomy of the holotype, so that original specimen that was used to describe the species, looks as though it might be a developmental abnormality. So I have always questioned
Okay.
whether or not Hafa Hafa is a real thing. And it could be. Oh, there is a second feature that really characterizes Hafa Hafa, which is that it has enormous scales on the occipital lobes. Right, it has a few very, very large scales. So it could be that that original specimen was just a weirdo, but it could also be that it's a genuine species. People claim to have found multiple different specimens and to now be doing kind of monitoring things, but I don't know more detail than that. So I'm a little skeptical about it. But it could be that just by chance, those features might not have genetically, like morphologically identified it, but it could be that just by chance it happens to also have been a distinct species that was described.
Hey, Mark, I want to say all these species we're talking about, you're saying, and then there's these little enclaves, the little areas where these species come from. And you had a, like, Brevicorn was just in a small area. Did I
Well,
see
it's
that
only
correct?
genetically sequenced from that small area, but Brevicorn itself is very widespread.
Okay. All right. I'm
Yeah,
just wondering because I mean, when I went, I saw Crypticum all over the place.
yeah, Crypticum is really widespread.
We stopped stopping the bus when
Yeah,
anybody saw Crypticum.
yeah, yeah, yeah.
And it was just, I was just thinking, okay, why is it that there's no quota for Crypticum? There's quota for Brevicorn. And I'm just wondering,
That's a good
did
question.
did that exist? Did Crypticum exist back when CITES was figuring things out, or do they think it was all Brevicorn?
Well, okay, there are two levels here because there is a Malagasy level and the CITES level, right? So the Malagasy government themselves also sets quotas, and the CITES quotas and the Malagasy quotas somehow correspond to one another. Uh, I think that possibly the reason that Crypticum might not have made it onto the CITES, and you'd have to look this up in order to know for sure, but it could be because it's found in Ambui Tantili, which is a very, very threatened area, and they want to avoid putting pressure on that area, and so they wouldn't put a species that's found there onto the list.
Okay.
But that would have stemmed from those early days when the species was poorly, 2006, 2007, 2008, when this species was relatively poorly known. And I guess that was in the process of when Crypticum was starting to be the name used for the rest of the range. I should mention, in the original description by Chris and Ron of Kalama Crypticum, they gave it this distinguishing feature of a rostral appendage that doesn't exceed 2.5 millimeters. But in the map and in the rest of the paper, they refer all of the populations from the eastern mountain chain, south of Andasi Bay, essentially. All of them are referred to Crypticum. All of them have rostral appendages longer than 2.5 millimeters.
Okay.
So they were using that as a diagnostic feature when it is clearly not a diagnostic feature, and that they were aware that it was not a diagnostic feature. So I don't exactly know how that happened, um, but there are it's a discrepancy that these
Okay.
things happen. Taxonomic works are challenging to compose. It's hard to keep track of all these facts at the same time. But that is a source of discrepancy that doesn't make a lot of sense in that original paper. Just bear
Okay.
with me while I turn on my light as it slowly
Yeah.
goes to
Okay.
nighttime
Well,
here.
let's do a very quick. Anyway, the question is, I know it's late there. How much more time do we have with you?
I have at least another hour if you're willing, but it's up to you, Bill.
I
So
am willing, but I know what
okay.
happens when you start listing species off one by one, and I want to
Right,
make sure.
exactly. Right,
So
right.
I want to be respectful of your time.
No problem. My
Okay.
my
Okay.
wife and son are asleep, so I think we're okay. Uh, so so we've gotten through 4 of our uh 10, 12. 4 of the 12 species. We're 1/3 of the way through the group. The rest of them go comparatively quickly. Okay. So our next species is Heleniocy, which I believe,
Yeah.
no, it was not the question. Peltierorum was the question. We'll come to Peltierorum.
Okay.
So Heleniocy is, so it's named after this person called Hellenius. And Heleniocy is a high elevation endemic. It's found on 2 tiny mountains called Ankaratra and Andringitra. And these 2 areas have very, very high microendemism. So microendemism is basically a word to describe it when you have species that are only found in a very specific single location, right? So both Andringitra and Ankaratra have species that are only found on those mountains. And then there are a couple of species that are found on both of them. They are quite high peaks, some of the highest areas in the entire east or central east of, or I should say central Madagascar. They are west of the main rainforest chain. Okay. Um, and so Ankaratra and Andringitra have this weird species. And the thing that characterizes Heleniocy is that in males, they have a bright red rostral appendage. In basically every other aspect. Oh, and it's very short. It doesn't really exceed
Okay.
the front of the snout.
The red seems to be, we got that in Crypticum and Brevicorn.
Yes, right. But here we have the difference in snout length, right? So
Okay.
whereas you would have a bright red rostral appendage in Brevicorn very frequently, it doesn't exceed it exceeds the snout by quite a long distance in males, whereas in Heleniocy it's very short and it's only found here. And it's also very unlikely that anybody for the pet trade would be collecting from these 2 locations because they're fairly inaccessible and they're fairly strictly protected. Just bear with me 1 second while I dive under.
Yeah.
I there we
Wow.
go. I'm back. So okay. Um, so yes, we are with Heleniocy. It is a mm, you know that's kind of the main distinguishing feature. There are probably some other scale counts and things that would also help. It's also fairly small. So males uh, are only 7 centimeters long. So that's a relatively small species compared to Brevicorn, which can get really beefy, much larger, right? So but in general, this thing looks exactly the same, and you could easily confuse females with females of the other species. They just might be a bit smaller. The next one is this thing called Calumma tsaratananense. This species is very poorly known. Tsaratanana is Madagascar's highest mountain, and it is found in the far north of the mountain. So basically, if you go up to the very tippy top of Madagascar, that's Montagne d'Ombre, and then you go down a tidgy bit, then there's a big gap. So Montagne d'Ombre, mountain. Tsaratanana, mountain, much higher, almost twice as high. In between them, there is a giant rift. So it's almost like they're 2 different continental landmasses. And basically, nothing found in Montagne d'Ombre is also found on Tsaratanana. But Tsaratanana is connected through mountains and rainforest with all of the other places that you might have heard of. Manangarivo goes down to Be Magnifica, goes across to Marojeji, Anzanahari Bay, Ambulukupatrika, Makira, and then all of that mountain chain, that's the kind of northeastern diagonal mountain block at the top of Madagascar. You go south from there into the pieces, so Makira down into Zahamina and then Andasi Bay and those kinds of places, right? So those are the main axes of kind of rainforest in Madagascar. So in principle, at least before all of this deforestation became such a huge problem, things that are in Tsaratanana could have been, would have been in connection geographically, biogeographically, with species that were found elsewhere, right? Or with areas elsewhere. And that is the reason that we have quite a lot of species that are found in Tsaratanana that are also found in Marajeji, for instance. And we have really interesting elevational structure there. So Tsaratananense is the smallest of the members of the Brevicorn species group. The only female that was studied in any detail in the Glavenensis field guide was just 44 millimeters long. So that's a female member of this group who is 44 millimeters is like this. So that's a really, really small member of this group. Now that might not be mature, but we really know very, very little of this species. And there are a few other distinguishing features, but it's such a rare species, only found at very high elevation. It's unlikely that it would kind of have any substantial presence in the pet trade. Most people will not see it. There are, to my knowledge, no photos of a live individual, although that might have changed with some of the subsequent expeditions to Tsaratanana. Very hard place to get to. You have to bring a white chicken to be sacrificed at the top of the mountain. And there are people there with guns who are protecting their marijuana fields. So you kind of want to avoid Tsaratanana, although the diversity of animals that are found there, I've described numerous species that were collected on expeditions there. It's a very, very cool area. Okay. And then we have this thing called Calama Peltierorum. Okay. So Calama Peltierorum was described at the same time as Calama Crypticum, for instance. There were 6 species described in this paper by Chris Raxworthy and Ron Nussbaum. And Calama Peltierorum was a huge mystery to me. I did not understand at all what on earth this animal was supposed to look like. And it was, described in the field guide of Frank Glav and Miguel Vences as being similar to Maita, but then not a lot more detail. I mean, they give some distinguishing features there. But in the original description of this species, Peltierorum, it has, and this is a really important feature, if you go to that original description, if you can find access to it, there are large yellow ovals on the flank of this animal. This is really important and something that I had not understood when I went to that area of Madagascar. And I found a lot of animals that, perfect. So this is a photo of Calama Peltierorum taken by Patrick Andriamihaza. So he has been up in that area with tours. And so when I was in that area in 2000, so this is, again, Tsaratanana area, right? Just lower down on this same mountain chain. So Tsaratananense is found very high up. As you get lower down, you have these weird animals that are large. They look, to my mind, it's really funny because Chris and Ron, in their description, they said that Peltierorum is found in the same places in this area as, in their paper, they said Calama Crypticum is found up there too. But this thing looks a lot like Crypticum. And when I was collecting individuals in that area, I thought I was finding Crypticum, and so I called them Crypticum. But it turned out that what I was finding was a combination of Maita and Peltierorum in the same place. Actually, on the same tree, you would have both of these species sitting side by side, right? Or at least extremely close trees. Okay, so this totally threw me for a loop. I utterly misunderstood what it was that I was looking at while I was going around there. But it turns out that uh, Patrick and I have both had the same experience, which is that we have both been to an area. This is close to B. magnifica, if you've ever heard of that. It has some of the rarest ducks in the world. But we've been to this area and we have come across this species, which looks for all the world like somewhere between Calumma crypticum and Calumma maita. And as soon as I saw and realized that the flank here. So let me show you. I'm going to change my background again. So this right here, I don't know if you can see that.
Let's see if I can.
This is a female of that species. This is a female Calumma peltierorum, and she's actually gravid. So she's got a beautiful bright red, or well, it's not very red, but there's a slightly reddish line around the eye, and this is I think a gravid female of that species. So this is Calumma peltierorum, and oh, Patrick's in the chat. Thank you for showing the picture. So Patrick took these beautiful photos, and I also managed to get a couple pictures. I'm just going to try and find a picture of a male, if I can. Do. And I didn't realize what I was looking at, right? So I was kind of collecting all these things under the same name, and only later did it become clear to me. In fact, it was when I saw Patrick's photos, and I realized, oh, this thing, it really has consistently this bright yellow marking on its flank. It was at that point that I went back to the original literature, and I was like, oh, right, what we've been looking at this whole time, all these animals that we were collecting, they were actually exactly this species, matching perfectly the original description of Calumma peltierorum, which was a bit of a light bulb moment. So here, let me use another. This here, this is a male. You see the bright yellow spot right in the middle of the flank? That is the typical feature that identifies the species of Calumma peltierorum. All right. So that's really nice. It's consistently there. You can see from that female that the identity of females is just as troublesome as ever. They all look exactly the same across all these different species, even through different sizes. They are often green or brown and have these pairs of white spots with the black dots on their flank, and otherwise they basically look the same. But this is a very typical male of Calumma peltierorum. Now, sometimes Peltierorum have a longer rostral appendage than this one. This is not a super long one, but they always have that yellow flank patch. And this is not even a very excited male. He was sleeping, or he might not have been sleeping, but he's certainly not fired up. So yeah, look for the yellow patch, and then you can say this is Peltierorum. But otherwise, it's challenging.
Yeah, and there's another of Patrick's pictures of a Peltiorum.
Exactly. Yeah, so you can see that rostral appendage. I mean, you wouldn't be able to tell that it's a different thing. Okay, so that's Peltierorum. It was a real mystery to me, and it actually brings us perfectly on to the next animal on our list, which is Calumma maita. So, do we want to take a brief pause before we go to Calumma maita, because it's going to be a longer one? And
Okay.
see if there are any more questions. I don't think we've missed any questions actually in the chat.
Oh,
So
there's
that's
that one.
well, yeah. Has facultative parthenogenesis been documented in any chameleon species? Not to my knowledge.
And what is?
Facultative parthenogenesis. It's all the rage. It was just documented for the very first time in a crocodilian, which is a huge deal. So, facultative parthenogenesis, we'll break it down. Facultative means when you wanna, if you need to, if you've gotta. Facultative is basically can, but needn't. Right? You can do something, but you needn't do it. That is a facultative thing, right? I am a facultative coffee drinker. I drink coffee, but I do not need to drink coffee. Um, unlike, for instance, many relatives of mine. Okay, so facultative. Parthenogenesis. Parthenogenesis is the fancy long word that we use for, let's say, unassisted reproduction on behalf of a female. So the female reproduces all by her lonesome. This is very easy for many reptiles because many reptiles have ZW chromosomes, not XY chromosomes, ZW sex chromosomes, where in humans and many other mammals, it's XY in males and XX in females. XY, we call that heterogametic. And XX we call homogametic, right? Same gametes, different gametes, right? In reptiles, in squamates that have this ZZ, they have a ZZZW system where the female has ZW, heterogametic, and the male has ZZ, homogametic. What that means is that the female can do, she could in principle, whereas females of our species only ever produce gametes that are X's, right? They cannot produce, at least if they have the most common chromosomal arrangement, they will only have two X chromosomes and they can only make X gametes, right? So it is the gamete that comes from the sperm that determines the sex, essentially, because it will either be the same or different, and therefore male or female. In squamates, it's the other way around. It's the female side of things that is determining the sex. Some of them are male, some of them are female, unless they have temperature determined sex determination, or they go to XY, which they can do as well. So it's crazy. Chromosome evolution, sex chromosome evolution is bonkers and a very active field of research. All right. So, but it is in principle possible for a female to self-fertilize and produce either ZZ or ZW ova. And so in principle, a female could give rise to male or female offspring on her own. And so there is this idea, right, that this is kind of a thing that many different groups of reptiles can do. I use reptiles here intentionally as the word. Birds are also reptiles, right? So birds, it turns out, can do this too. I don't know if you heard about the California condor that also had a virgin birth recently, or a virgin ova, I think. I believe that was the documented case, that it was a California condor. Crocodilians can do it. Squamates can do it. I don't know if rhynchocephalians, the tuatara, I don't know if it's been documented in them, but there are many, many, many different groups that can do this. It's in many cases, it is what we call facultative. So, for instance, let's say you keep a rattlesnake all by itself for a very long time. Well, at some point, that clock inside the rattlesnake, the female rattlesnake, might start clicking, and it actually might figure out by chance that it can do this thing where it produces fertile offspring, fertile eggs all by itself, and those eggs can hatch. And that has happened in captivity, especially with squamates, so lizards, including snakes. Snakes are also lizards. Um,
Okay.
all of those things have happened, fairly frequently, right? Okay, so does that? I guess that answers the question, right?
Yeah.
We haven't had it documented in chameleons.
Okay.
That does not mean that it's not possible. In fact, I think it's highly likely that it's possible.
What? Okay. Well, I know this is getting way off the topic, but why would you say that? Wouldn't we have seen that or witnessed it?
Because there is this really annoying thing called sperm retention, where you might have a female who has at one point been for 30 seconds together in the same cage as a male, and you didn't see it happen. Nobody saw it happen, but she might have stolen some sperm and then kept them for
Yes.
like 8 years. So, um,
But we are captive.
when we are captive breeding ourselves and we keep the females exclusively, completely away from other males, it has not been documented as far as I'm aware, but it also is ultra rare in the species where it does happen. So it doesn't seem to be something that they just turn on by virtue of being isolated and alone for a long time. There seem to be, um, we might call them Goldilocks conditions that lead to this kind of
Okay.
spontaneous thing happening. So it's not every um, Komodo dragon that does it. Not every Komodo dragon, if you keep her alone, will produce eggs on her own, but some will sometimes. Same is true of pythons, of rattlesnakes, of lizards of various kinds. So yeah, I think that is like the gist of things. It simply
Okay.
hasn't been observed yet, or hasn't been documented or confirmed yet. And once it happens, there is a whole rigmarole that you have to go through in order to confirm that it has indeed happened, because we have to basically prove that you didn't miss something, that a
Yes.
male
Yes.
didn't break out of its cage and climb in with the female or something, right? So, and that's expensive and hard to do, where you have to prove that this thing is genetically identical to the female, and would have then basically identical copies of every gene across the whole genome, right? And
Okay.
that's not super easy to do.
All right.
Okay, so we can move on to the
Move on.
Calamamate species group, or species complex. Okay, so Calamamate, as I mentioned already, but for those of you who have joined us midway through, is extremely widespread. It might be, within rainforests, Madagascar's most widespread species. I'm saying that, and at the same time trying to reassess. Other potential candidates for that title would be Firstifer wilsey, immensely widespread, um, but not much else. So, Calamamate is a beautiful species that looks quite a lot like this chap up here. I guess, Bill, you're in the process of maybe pulling up a picture, but I can also do the same thing.
Okay.
Um, because on the same trip where I was finding these stupid Peltierorum and getting very confused by them, I was, of course, also finding Calamamite.
Okay.
I think this is one. Wait a second. I can't see the picture very large on my computer before I show it to you. Yeah, so this is Calamamite,
That doesn't
which
look
is,
like the one we expect it to look
it
like.
doesn't look like what you expect it to look like. And that's because Calamamite has a lot of geographic variation in its appearance. Let me just see if I can find another picture from the same place that is slightly more indicative. Bear with me. And so, basically, as I mentioned before, here's a more typical one. Okay. So,
Let's see. I can turn this on. This is from our good buddies at MadCam, and so
right,
that's what we're
right.
used to.
So that's what you're used to. You're used to Kalamamaita having these beautiful green occipital lobes that are very, very long. You see how long, Bill, if you go back 2 slides, maybe one more. Yeah. So you see how long those occipital lobes are? The angle of them, relatively flat, enormous surface area of the lobes, which of course those lobes have muscle attachments and they flap them up and they hiss at you in order to kind of threaten you, right?
Yep. Yep.
So they also have that very substantial rostral appendage that you can see there, and you can also see on this individual behind me, maybe. And so they are found, as I say, very, very widespread, but with some color variation. So those things that are found there um, are found in I think those will have been observed in eastern Madagascar, maybe down into Amasina, or even in Andasi Bay. I think they occur. So you've got a very, very widespread species that comes in kind of a mixture of different appearances. And this one that I have behind me, these are these really interesting northwestern individuals. So this is from the area of close to Tsaratanana. So that area in northern Madagascar, if you imagine Madagascar as the shape of my hand, you would have, I don't know how to make it more like Madagascar, but yeah, we can kind of gently do that, right? There we go. So if this is Montana Dombra up here, then this is kind of Tsaratanana, and that's where this guy's from. You go across to the east. I'm having to do this mirrored on my hand, which is quite funny. If you go across to the east, now we have Marajeji. Marajeji also has Maita, but the Maita there look also different. I'll pull up a picture in a second. And then you go south, 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 south. And as you go south, you go into these populations that have that very distinctive green coloration. They even have dark dots on the side. And so it could be that this guy here is a different species that is awaiting description. But I don't have strong genetic evidence for that yet, let's say. It is a project that is in need of work, but there are many, many, many projects in need of work in Madagascar. And as you go further south, there's more variation. And again, we have this problem of females, even females of Maita, they're more easy to distinguish from the other members of this species group than they might be, but they are pretty challenging things to identify still. Okay, so that's Kalama Maita. They're absolutely beautiful animals. They are, yeah, as members of this group, they're very distinct. But now we come to this problem. They're actually not members of this group.
What?
Hey. Okay, so now we turn back the clock to the year 2013, where the last decent phylogeny of chameleons was published. Decent evolutionary tree of chameleons was published. And in that study, they showed that Kalama Maita is one of the deepest genetic lineages within a group that is comprised of the Kalama Brevicorn group and the Kalama Nasutum species complex, including Gallus. Okay, so it turns out that, at least based on their analysis, which you should bear in mind is not based on a huge number of genes and is subject to change, that story is for the future. Okay, so there are essentially 4 different species groups of Kalama, right? There is the Parsoni species group, gigantic animals with very few exceptions, comprised of O'Shaughnessy, Globifer, Umbriensis, Parsoni, and Caparoni is close to them. We'll get to Caparoni in a second as well. So Caparoni has these large occipital lobes, looks somewhere in between. It turns out it's closely related to Parsoni. So they're over there. There's also the Kalama furcifer species group, which is a really frustrating name, but there's a species of Kalama called Kalama furcifer, because furcifer means forked, and it has a forked rostral appendage, so it is an appropriate name. It's just unfortunate that there's an entire genus called furcifer too. So the Caloma furcifer species group, which includes things like Caloma gastrotenia, Caloma tarzan, Caloma vensizi, Caloma glavi, these things that are all green, very homogeneous species. And then there is this other big group that is divided into two. One is the Nasutum complex, and one is the Brevicorn species complex. And you would have thought that those are two nicely separated little units, but they're not. What has happened is there are two things that are
Wait,
problematic here,
which
if I
units?
keep
You
my
say
language clean.
Nasutum and Brevicorn?
The Nasutum complex is closely related to the Brevicorn complex. Yeah,
No.
they're actually sister to one another. And not only are they sister to one another, Maita sits outside them. Maita is sister to the pair of them. So that means that Calumma Nasutum is closer related to Calumma brevicorn than Maita is. It gets worse. It gets worse.
Oh, great.
Calumma cuculatum,
Yes.
which is very much like Calumma Maita, for instance, according to their tree, is more closely related to Calumma gastrotenia.
Okay.
Yeah, exactly. That's how everybody feels. So we can just pretend that this is not real. As I say, the tree, it was 2013. It was really, really good genetic representation for the time. And there has not been anything published since that is better.
Okay.
But that's mostly my fault. Because and well, it's actually the fault of various extremely mean reviewers, but we'll get to that at another time. But in essence, right, there is a problem where Maite does not fall deeply in this group. Cuculatum does not fall deeply in this group. Caparoni does not fall deeply in this group. So something is really, really weird going on here. But for our purposes, in the happy, cushy
By the way,
world of
you know what?
keeping chameleons,
I'm just going to bring up. Let's just show everybody what cuculatum is.
yeah, let's look at Cuculatum. It's a weirdo.
Just
But
so
I think nobody looking at this animal is ever going to say, "Oh yeah, that looks a lot like a member of the Gastrotenia species group."
yeah, that's so. Anyway,
Right. So, yes. So that's Maita, right? It causes evolutionary problems. It is diverse, widespread, and potentially more species than one, let's say. I think that there were possibly yeah, so Maita was used as the outgroup for the Boumans et al. Study that I mentioned before, and so we don't know very much about it in terms of its genetic variability, but that's work to be done, let's say. Okay, so we can move on from Maita and talk actually about Cuculatum, which is the next here on the list. So Cuculatum is that weirdo that Bill just showed. I haven't had a chance to see one in life. They have a really wide snout, which I think is fascinating, and a very narrow body. Actually, whenever you see a picture of them, you can identify them instantly by the head shape. You see how long the head is? It's got those occipital lobes are going forever, and it has this bright white stripe down the side, and that is almost always present, except when the animals are really, really stressed out. So you can, if you see a cuculatum, it's a recognized on-site species. It's basically the only recognized on-site species that we'll talk about today, because it is very, very, very hard to ID, or very, very hard to confuse with anything else. Sorry, so everything else is hard to ID, and then you have this thing that is just like it has orange eyes. It's often got orange on that rostral appendage as well. So you know, we don't really have to talk about it any further, but it's super widespread. But as I mentioned, it's usually in low densities. Okay. And then we have just a couple more. The first one is Kalamajeji. And Kalamajeji is a high mountain endemic. It is considered one of the most threatened chameleons in Madagascar because it's only known from the peak of Marajeji. Jeji Marajeji. Right.
Okay.
So or Marojeji if you want to say the Malagasy way. So up at the top of Marajeji is an extremely weird habitat. It is, you leave the trees, you're above the tree line, and you come to this open, marshy grassland. It is truly one of the most spectacular places I have ever been, and I will try and put up a background while I'm talking, just to give you an idea of what this place looks like. Now, there have been attempts to figure out like what is going on um, with this population because uh, right, you're above the tree line, which means you start to get worried about things like fire, um, and especially as droughts continue. So here, this is kind of the split up 1 second. So this is the type of habitat that we're talking about. That's, by the way, this is Miguel Vences, and this is Molly Bletz. You should know Miguel by now. He's the one who co-wrote the field guide. And Molly, she works on frog fungal diseases, basically. So she works on chytrid, which is this fungus that's eradicating frogs globally, which is a huge problem. Right. So this is the type of habitat that we're talking about for this chameleon. There are bushes, lots of bushes, but and these marshy, weird wetland areas. There's essentially no algae growing in this, or very little to be seen anyway. Totally strange habitat. It felt like an alien world to be up there. Absolutely beautiful place to be. And the concern was, after multiple expeditions, we went up there for a single day. It searched intensely with some of the best chameleon finders in the world. We could not find individuals of this species. And that made us worried that potentially Kalamajeji could be not there, right? It could have gone extinct without us noticing. Because there's this thing that happens at the top of a mountain. You have a mountain, and you have a species that's only found here. And now there is a thermal area around this thing, up and down from that point, that is the thermal tolerance of your species. Now, that thermal tolerance, that changes quite slowly, evolutionarily, millions of years potentially. But climate change changes fast, very fast, especially with humans involved. So, if you have a global climate change of 1.5 degrees, that could be the difference between here and here. But there's no mountain here anymore. And when you reach this point, that's when the species goes extinct. If your species can no longer survive at the habitat that it's living on, it's been pushed off the top of the mountain. And when it gets pushed off the top of the mountain, it goes extinct, right? So that is the concern for any mountain endemic. And anything living in this habitat is at risk of that. They have to adapt very fast or go extinct. That's basically your options. But extinction is a natural process. This happens all the time. So
Is there a name for this?
I don't actually know.
Okay.
I guess it's called mountain peak effect if
Yeah,
we're
I
going to be
guess
naming it. But
we've come to a point where we better come up with a name for something like this.
I guess there must be a name for it, but I don't actually know one.
We've never had to have a name for it before.
Yeah, yeah. So this effect causes major concern. There's a second species of chameleon that's also found here. In fact, there's a third species of chameleon. The second species of chameleon that's endemic to this area and another area not too far away is called what's it called? I can't remember. Kalama. Wait for it. Wait for it. I can't remember what it's called. Kalama. It's the one that caused all the problems. Bill, I sent you pictures of it.
Paraname?
Peyurazi, that's the one. Yes,
Okay.
Peyurazi. So Kalama peyurazi is also found in exactly this habitat. The thing is that Kalama peyurazi is also found on another mountain, but Jeji is only found here. Okay. So we were very worried about this. The good news is it has been found since. They are
Okay.
there. And in fact, the people who found them said that they are found in moderately high abundance. So you just have to know where to look. And apparently we didn't know where to look.
Okay.
Good to know. So all of that means we don't have to be as concerned, but we should still be actively kind of monitoring these populations. You do not want to do active monitoring in this place because it's like it can be a beautiful open 1 second and then completely clouded and thunderstorms another second, and you're in the lightning, right? You're in the cloud, so it's yeah, not a super spectacularly nice place to be when the weather is inclement, which it is often.
Okay.
Okay. And that brings us basically to the last member of the group, which is Kalama Tsikorn. Before I go on to Tsikorn, I'll just maybe address this question from Melanie. Are the cyclones contributing to their possible extinctions? Yes, Cyclone Freddy was catastrophic, and yes, cyclones are a major, major, major risk. And of course, cyclone frequency and the strength of cyclones. I don't know if you guys saw what happened with some of the recent cyclones, but they went over the island, turned around, came back, and then went turn around and came back again. So
Wow.
they got hit 3 times by the same cyclone, which has never happened before. And sometimes those cyclones are going over Madagascar and keeping strong enough that they slam into Mozambique and are causing flooding in Mozambique and Malawi, which is just insane. That level of power is just unbelievable. And yeah, it's a major consequence or a major problem for these areas. For the animals themselves, at this point here, I wouldn't be majorly concerned about being in a cyclone because other than the electricity, um, uh, because there's no trees to fall on you. Right there, the worst that happens is that your bush gets blown over and you're not falling very far. So, from that perspective, I guess, you know, if it weren't for the electricity, I wouldn't mind being up here when it's that stormy.
And by
But
electricity, you mean lightning?
lightning, yeah, yeah. I have nearly been hit by lightning several times in Madagascar, and I know someone who's been hit by lightning 3 times. So, yeah, yeah, it's not something that you want, certainly. Okay, so the last species is this relatively I say relatively, extremely poorly known species found in the very far southeast of the Madagascar called Kalama Tsikorn. Um, there are a handful of individuals that are known of this species. It's from Anduahila. So Anduahila National Park is a 3 parcel national park. So three areas of forest that are protected. They have different bio zones within them. Um, it is true rainforest at the far southeastern tip of the island, but on the west-hand side, one of the parcels actually has transitional forest between rainforest and spiny forest, which is the opposite end of the spectrum when it goes to moisture. Spiny forest is crazy dry, right? So sorry about the sniffing. I am recovering from both allergies and a cold. So
Okay.
yeah. Anyway, so Kalama Tsikorn is only found in that area, and I don't know if any tourist has ever taken pictures of one. I'm not aware of any photos on iNaturalist, although if I'm honest, I have not checked recently. Um, but yeah, it is a species that's, again, it's never going to come into the pet trade. Um, I think that it would be, yeah, 0 observations in INAT, with good reason. It was originally described also by Chris Rexworthy and Ron Nussbaum in the same paper together with these other ones. And, it's identified, it's called Tsikorn. Tsikorn, so Tsi is the Malagasy word for no. It is the negation, Malagasy negation. Um, so tsimisi is there is none, right? Misi is there is some. Huh? So tsikorn is a portmanteau or a very weird combination of Malagasy word tsi and the Latin word corne, which means horn. And it
Okay.
basically just refers to the fact that this thing does not have a rostral appendage in males. But other than that, and the original description and the original specimens, there is basically nothing on this species. So we don't even have genetics on it. So it's a big open question mark.
All right. So I would like to step back and ask the question. We have Brevicorn complex all over the place and all these different species, Nasutums all over the place, all these different species. Why are they doing that? Well, Pardalis is all over the place, but they're just different colors. And then Ustaleti is all over the place, and it's the same species. What are the dynamics between these groups?
Um, yeah, that's I
And I
guess it's a
realize you may not be the
fundamental
one
question.
responsible for this.
Well, no, it's not that anybody's responsible for it. It's that evolution um, doesn't always do the same thing with the same substrate, and we're not working with the same substrate. We're working with lots of other different things, right? We are working um, with a huge diversity. Sorry, I'll put my angry guy
Yeah.
back.
And
Um,
I mean, maybe the question is, how come Ustaleti is so homogenous, do you say, or the same?
yeah, so I guess it's a combination of things, right? So on the one hand, there is the opportunity for the arisal um, of differentiation, and that can happen under different circumstances. So climate change can cause populations to retract into tiny little pockets. Um, if they're in those pockets for long enough, they might generate differentiation and expand outward. And then when they meet again, there's no contact, there's no gene flow. And that process might be more extreme where mountains are more prevalent. And now all of these rainforest species that we're talking about, they're found in pretty mountainous areas. Whereas Ustaleti is found mostly in lowlands. Okay, I give you all of highlands of Madagascar is hilly, very hilly, but it's not maybe what you might call mountainous.
Okay.
There are also these big drainage basins, and these things are found across enormous drainage basins. I mean, the water that is in Antananarivo, everything west of the big mountain chain, that all of that water flows out of just a couple of rivers. So there's opportunity if things get on a raft and raft down the river, they kind of spread, or they basically would all converge. Anything in all of the water that falls in Antananarivo, all the way up to Bay Magnifica and Saratanana on the south edge of that thing. All of that water goes to the Betsibuka, all of it. So you have enormous amounts of movement within a single geographic unit. These drainage basins, they act as units. In the U.S., this is relatively well known as well, because how much of the U.S.'s water goes out the Mississippi River? Most of it, right?
Okay.
So you have that process, and that actually leads to some biogeographical connections between these different things. And so on the one hand, it might be that climate change, these retreat expansion, retreat expansion, these things, they could get in the way of gene flow. You can also have rivers of substantial size, where on the west hand, on the west coast, like there are really big rivers, but if you go far enough, you can far enough upland. You don't have to go very far up. You just have to go very far distance wise. You can get across those headwaters. Whereas on the east coast, if you're east of the mountain chain, the rivers are very wide, very fast, because the Madagascar is shaped like this, where there is a whole long, long slope to get to the seaside the sea on the west side, but a very steep slope on the east side. And those steep slopes, they're bigger rivers earlier, and that can also be big barriers to gene flow. the map of species differentiation in Madagascar doesn't really match the rivers very well. So it's kind of an iffy thing, especially in the lowlands when you're talking about places like Tuamasina. There you might expect those big rivers to be getting in the way. They certainly block human traffic. I mean, I don't know if you guys had to wait for your whatever they're called, barge, to get across some of the rivers on your travel up to the northeast, on your way to Marancetra. But certainly those areas are almost impenetrable to um, to humans in the lowlands. So
Okay.
chameleons aren't crossing those things either, right? Or very, very seldom. So there are those factors involved, but then there are behavioral things. Maybe it's that these species in Columba, maybe they're just more likely to evolve characters that are preventing them from isolating from one another faster than, for instance, Fursa ustelleti. And then there is this big thing called vigility. It almost sounds like a bad word, right? Vigility. So vigility is the properties of the dispersal rate of a species. It's essentially the question of how much does this species move on average per year, per lifetime, per generation, right? And if you have a very vagile species, this is where it starts to sound dirty. If you have a very vagile species like Ustelleti, very capable of covering large distances, you see them running across the road.
Yeah.
They do not hesitate to go onto the ground to cover distance because they're in arid habitat where the trees are not side by side. Vigility in that species is very high. But you go over to a species of similar size, let's say, for instance, Columba. Well, the best one is, of course, Columba parsoni.
Yeah.
Columba parsoni, that's a big chameleon. But how often do you see them crossing a road?
I,
Basically, never. They will very, very, very rarely come down from the tree. The level of vigility is small. Vigility is very strongly correlated with gene flow, because if you're happy to run across the road, you might as well mate with things on the other side of the road. If you're not happy to cross the road, you might not want to mate with things on the other side of the road, and as a consequence, your genes don't get spread to the other side. So vigility is also extremely correlated with body size, because you can do all the running in the world, but if you're only this big,
yes.
that running is not going to get you very far.
Yes.
But if you're the world's longest chameleon, that running is getting you a hell of a distance, right? And that's the other axis that we're talking about. Obviously, there are very large Columba species, but those large Columba species they're still not very vagile. But Nasutum, the part of the reason the Nasutum complex has been so divided, or needed to be divided so extremely, is that those are small chameleons that have very small genetic units, right? All right, so you're pulling up the slide, right?
Yeah.
So
Yeah.
this is right. So this
Those
is
are
the
in the
Columba
student.
Nasutum species group, if we say in the broadest sense of the word. On the top left there, that looks like that's actually Columba rhodomanos. There. So rhodomanos has the integrated rostral appendage with the first scale on the lip. And then the second one there, that's
That's a
the middle
linotum.
on the top row. That's Columba linotum,
Yeah.
for sure. You can see the nice rings on the tail, the blue flaps on the ear, and the blue rostral appendage. Very nice little juvenile there. And then top right, you have what could, you might think this is a weird Columba nasutum, but actually this is a female of Columba gallus, and male Columba gallus is on the bottom right hand side. And then on the left hand side, I wasn't sure before when I was talking with Bill about this one. It's either Columba phallax or it could be Columba tiasmantoi, but it's very hard to tell in this individual in these colors. I would lean toward phallax if I have to take a guess. But those species are sometimes challenging to tell apart. So Right. So the Columba Nasutum group, you can see there in the top left picture, that's a large individual for that species group, right? They're tiny little lizards. And yes, they can cover a substantial different distance during the day. You know, they move multiple meters during a day on their prowling because they are, like all chameleons, they are cruise foragers, right? Or most chameleons are cruise foragers, which is the technical term for when you don't sit still, you just move around and
Mhm.
hunt for food. Um, right. So these animals are are very small, and their distribution range is often extremely small. So we have tiny microendemic species. Calama Yuliae, for instance, a species we described from a forest. When you drive toward Andasi Bay, you drive past a tiny, tiny, tiny parcel of forest where it just so happens that David Pritzer and Frank Glav and a team of people, they got out together. They went and looked inside this forest, and they just happened to find an individual of the Columba. Um, it's actually from the Columba Nasutum Columba Butkeri species complex. Sorry, the Columba Linotum Columba Butkeri species complex. This group that have occipital lobes again, right? And they happened to find an individual there. It happened to be an undescribed species. They named it after David's wife, Yuli, so it became Kaloma Yuliae.
Okay.
And so Kalama Yuliae, found in that tiny forest, has been found in a single other location since then. But in essence, it's a very, very microendemic species. Kalama rualuku, this is a species that's causing me a lot of concern right now, because Kalama rualuku is found in we described that species as well. It's the smallest member of this species group. It was found originally only in forests south of Andasi Bay, but has since been found in Andasi Bay as well, and actually turns out to be a little bit more widespread. But it has entered the pet trade under the name Columba nazutum, and is being traded in, or actually, I don't know what species. It must be coming in as Bitgeri, or Linotum, but it's obviously neither of those. It's not found anywhere near those things, and it turns out to be, um, well, it's being imported in large enough numbers that that some people have been able to set up breeding colonies of that species, which I think is great. But it should never, ever, ever be coming into the pet trade, because there is no quota for it, and we have no idea how the population is doing. And only once those things are established should it be coming into the trade. And yeah, I mean,
Yeah, if anything comes in as Nasutum, Linotum, Butkeri, we don't trust any
yeah,
of
you
it.
can't trust it because the people can't ID them, and all the keys in the world wouldn't help them, because they're not even taught the technical identification things, and your average customs person just can't be expected to know that. That's fine, but it is a problem, and one of the reasons that increasingly over the last few years. So this is a very controversial take that I'm going to say here, but I've talked about it on my podcast before. Um, I increasingly lean toward this idea that we should have a green list for species being exported, and that would mean only species that are on that green list should be exported, and not everything is fine until you ban it. And
Isn't that what we have?
no, so the reason that chameleons cause problems, or the reason that chameleons are protected, is because all chameleons at genus level are protected, but any of the snakes from Madagascar, with exception of the boas, can be traded willy-nilly. There are no quotas for them because they're not CITES listed. So
Okay.
you
So
can
you're talking
actually
about
trade any of
outside of
them.
chameleons.
Right, right,
Okay.
right. And this would apply also in these cases where these animals, they are protected to some extent because they're protected at genus level. Um, but nobody, like the quota system is failing because nobody's able to identify them. So we should be screwing down, clamping down on that level of trading, where people are knowingly, they are certainly doing this knowingly, collecting individuals that are not allowed and exporting them into the pet trade. Like, until we described the species, Rualuku had not appeared in the pet trade to my knowledge, and now suddenly there are breeding colonies being set up, and
But
that must be
why
targeted collecting.
the only advantage of someone doing that is if that that location is closer to them than the Lenotum, Butgeri, or
which
Nasutum.
it possibly is. I mean, if your collectors are usually collecting in eastern Madagascar, then those things are there. Lenotum is in the far north, and much harder to get from
So,
someone based in Tana than these eastern things.
so did they become aware of the area where Rural Loco is because it was described?
Probably,
Okay.
yeah.
Because they're not getting any benefit for having this rare species if they're
Well,
sending
they
it out
might
as
be.
Butgeri.
They might actually be earning more money for it. Possibly not due to American clients, but clients in other parts of the world who are asking for it directly.
I see. Okay.
Yeah. So it's not entirely clear to me. We don't have numbers on it, and I'm not aware of anybody keeping an eye on it. But basically, it cannot be a coincidence that we went from this animal not appearing at all in the pet trade to suddenly large numbers, enough that people are assembling,
Could that
colonies, and it's
be
appearing by other people.
because we didn't know to look for it
No,
before?
no, it's so distinctive. It would have shown up in
Okay.
pictures for sure.
Okay.
Yeah, yeah. Because Ruuluku is really like it is two-toned, green and white,
Yeah.
super distinctive animal. And whenever we saw photos of it, because there were photos in the wild floating around online before we described it,
Okay.
so it was known about, but nobody had realized that it was a separate species. And it was only in the act of describing it that we kind of characterized exactly where it's found. We, of course, published exactly the location that it was found, and um, and made it known it's the smallest species, and blah blah blah. So and people do pay attention to those publications in terms of like being like, oh, that's a new place that I could go hunting. So
So, how do you protect an animal that you are describing before CITES catches up to it?
I am constantly worried about exactly that question because I don't think that we can right now. It's just not done. It's basically this is why I think everything should be CITES listed and then some things should be delisted, right?
Okay.
Rather than the other way around. The current policy is free trade in anything. This is country regulated at a different level. But in general, the global policy, if there are no other laws in place, is free trade in everything except the few things that we say there's no trade in, right?
Okay.
And it should be the opposite way around. And there was a paper by okay, so there's a podcast called Herpetological Highlights, which is Tom Major and Ben Marshall. And Ben published a paper. Ben and a team of other people, they published a paper on exactly this. That the current reptile trade, in particular, is essentially backward in the way that animals are being traded, and it's leading to a lot of trade in animals that we have no idea how threatened they are, or threatened species we know are threatened but haven't been CITES listed yet,
Okay.
which is, and every time you look at any of the CITES listing documents, if you go through and read those reports, they're always like, oh, this has been traded in huge numbers until we started to regulate the trade. This was also true, of course, of Bruchisia perarmata,
Okay.
was traded in massive numbers, and then we said no. And then Paradura masube, the same story. These geckos, huge eye geckos, traded in large numbers. Then the CITES thing came in and said fewer, right? And at the same time, the Malagasy government put in smaller quotas or zero quotas. So it should not be happening that way around, because then we can find out about the problems too late.
Yeah, that does make sense. And it would seem like if you're setting up a system like CITES, it would be.
Should be the other way around, right?
Yeah.
But I think it's harder to regulate, it's harder to control. And the other thing is, let's look at the Columba species. Even if you regulate it at the level of the genus and you say all Columba are by default CITES 2, which they are,
Okay.
it's obviously not working, because these animals are CITES 2. The same is true of Europlatus giganteos. There is a zero quota for giganteos, but they're still coming into the trade because they look exactly like Fimbriatus, and the people at the border can't tell, and they're easy to collect in the wild, and so you can easily sell Fimbriatus as Giganteos as Fimbriatus to your dealer, who then instantly IDs it by looking at the eyes and says, look, I have Giganteos and I can sell it as Giganteos and I can make more money because you're not supposed to have Giganteos in the trade. So these animals should also not come onto the public trade because they're illegally imported, but they still do. So
I see. Okay.
Yes. So yeah, is there anything else that we need to talk about the Brevicorn complex to bring things back here?
I think we have done a very good job
Very
covering
thorough,
it.
right?
And yeah, I think we've done a very thorough summary of it, and we've answered the questions. We've described the issue that we're all seeing and part of, and we've although there's no solution, at least now we all better understand what we're dealing with.
Right, exactly. I think to kind of wrap it up, I would say most of the diversity of the members of the Columba brevicorn species group are not things that we need to be worried about coming into the pet trade and being confused, right? Most of the species that I mentioned, most people won't have heard before.
Yeah.
And the only real ones that are coming in any likelihood in good numbers are Crypticum, Brevicorn, and Maita.
Yeah.
And Maita is easily identifiable or fairly easily identifiable. Females are sometimes a little iffy, but for the most part, females and males can be identified. Of course, if Maita is a species complex, that's its own problem. But Crypticum and Brevicorn remain the source of problem, and we do not have, until our exporters are bringing in all of their specimens with, and the importers as well, are getting those specimens with locality data, we will not easily be able to tell if we're bringing in Brevicorn or Crypticum. As soon as we have locality data, even relatively vague locality data, we would be able to say with some probability which species we're talking about, because they do not. There are no known locations where they co-occur at the moment. It's likely to change at some point, but for now, we don't know of any places where they co-occur. So, but
There's no
until that
places
point,
where Crypticum and Brevicorn
not that I'm aware
do
of.
not. Okay.
Yeah, but until that point, it's just going to be a matter of pairing them up and seeing what happens with the females, because the males
Well,
are, they're pretty indicative, right?
okay. So in this one right here, so that would mean this is either Crypticum or the first two
Uh-huh.
are, and the one down below, that's either Crypticum or Brevicorn, and we assuming that's Crypticum.
It's Crypticum. Yeah.
Okay, so those females, the first one and the bottom one would be Crypticum as well.
Yep.
Is that correct?
Yep.
Okay. So
as far as we know, genetically, nothing that has been found in Fianar has been assigned to Andasi Bay populations to the Brevicorn lineage. So we work on the assumption that we haven't missed them. Basically, they could be there and we're missing them. But at least for now, basically everything in Ranamafana, that's all thought to be Crypticum. That does not necessarily mean that that's correct. It's just based on the genetics that we have so far, which is a small sample because we're only allowed to collect so much.
okay. In that case, we will leave it at that. And I'll say thank you very much for coming on and sharing all this with us. Boy, there's a lot to sort through.
I'm sorry, it's an exhausting topic. Yeah, they're challenging creatures for sure. I think it's great that you've put together your guide. That if I can give one point of consolation, is that the habitat and ecology differences between things that are living in Andasi Bay and things that are living in Ranamafana are minimal, low. We have a lot of species that are found in both places. There are
Okay.
also a lot of species that are only found in one place or the other, but for the most part, there's pretty good continuity there. So unless the things that you're getting in are coming from the far south, which is very unlikely, you're likely to be dealing with animals that are moderately similar. So what goes for one probably goes for another. I would love to know actually of people who have experience keeping these things, if they keep their Brevicorn and their Crypticum separately, if they have that experience as well. Actually, if they find that the animals are liking the same kind of conditions.
We'll let you know as soon as we can. We'll be gathering as much information as we can, and we will probably all be sorting through it for a while.
Yeah,
Geez.
for sure.
All right, but would we? But would we expect the Crypticum to show blue legs eventually? You mentioned there's a population where it doesn't have blue legs.
I don't. Well, there are populations where I don't know if they would have blue legs.
Okay.
That's not the same thing, right? Yeah, work on the assumption that if it has blue legs, it's Crypticum. And if it's male
Right.
and it gets blue legs when it's fired up, then it's Crypticum. I think that's the safest assumption to make for now because that's what the evidence says at the moment.
Okay.
Yeah.
All right. Well, we have one question. We'll take one more question. Gabriel's
Some of
asking.
the
Let's
snouts
see.
of Calyptratus have bumps where maybe horns would have been. Is there anything to that? Is it just a personal observation? So no members of the genus Cameleo have horns, right? Just reviewing in my head if that's correct. I'm pretty sure that's correct. So
Yeah, no.
Camellia calyptratus, as of course the Yemen chameleon, and or veiled chameleon as it's sometimes called, and yeah, they might have slight, we could call them rostral protuberances or rostral prominences, but horns are not present on any members of that genus. Um, so the answer is essentially no, there is nothing to that to the best of my knowledge. However, horns must evolve from something at some point, so maybe they're in the process of evolving horns, and they haven't yet. And the horns of things like Trioceras jacksoni are keratinous and keratinous all the way through, so they don't have. Oh no, that's not true. Actually, they have bone underneath the horns, right? So yeah, it's a keratinous sheath around the bones.
Well, did we miss talking about Crypticum?
Yes, you missed about 2 hours of talking about Crypticum.
Fond boy, go ahead, start at the beginning and listen through it. We have been talking about Crypticum. We talk a lot about Crypticum.
Yeah.
So you have a lot of good Crypticum material to go through. We love Crypticum.
Absolutely.
Oh. All right, Mark, I'm going to send you to bed.
Thank you. Yes, it's
late
late.
there. Really appreciate you coming on, and have a good night, and we'll see you online.
My pleasure. Thank you so much for having me, and I hope you've enjoyed it. And if people have enjoyed it, you can follow me on social media. You can search for my name, it's there. I'm the only one, so you can find me very easily. All right.
Excellent. All right, everybody.
Thanks a lot.